Hey, good afternoon, everybody. It's my real pleasure to introduce Courtenay Ray, who is the first graduating PhD student out of my research group. Courtenay joined the lab in 2017 when I was at Arizona State University, and I was surprised I got her. She came with a lot of experience with birds and in the Pacific and has ended up working on plants in the Rockies. But seems to have thrived and enjoyed that work quite a lot, and has done some really great and really creative science in the process. Courtenay, well, you'll hear about her science. I won't say too much about that, but you may not know that Courtenay is also a banjo player. I think getting to be a pretty good one. And you may also not know that Courtenay is a big green chili lover, and between the two of us, we managed to almost torch several hundred plant samples in my car with a bunch of green chili steaming from plants during the field season a long time ago. But more broadly, I just want to say, Courtenay, you've been a great person to have in the field. You've been a great person to have taking care of our data sets, and a really creative scientist that I think has really pushed some of the directions of the lab in directions that it never would have gone otherwise. So thanks so much for being part of the group. I'm really excited to see your talk, and I'll let you show us all the great things you've been doing.
Thank you. I know most of you, but I still thought it would be worthwhile to start with a brief introduction of who I am. So my name is Courtenay Ray. I did my undergraduate at the University of Washington in geology and biology. I moved on to UC Santa Cruz, where I studied invasive species under the tutelage of Ingrid Parker. Then for my PhD, I joined Ben's lab at Arizona State University. And then sometime around January 2020, we transferred over to Cal. And a lot of in setting up this talk and thinking about my experiences during graduate school, I thought of kind of these really powerful landscapes, experiences of rainfall and experiences of being up on mountains. And also as a human, thinking about what that also could mean for the plants. So most of my research is done in this high Alpine plant community, Mount Baldy. And this research site was started in 2014 by Ben, and I joined this project and helped continue this research since 2018. And since that time, so this upcoming summer will be our 10th year of field research. We've seen a lot of different climate. So we've seen, for example, in my first year, June 6 in 2018, the field site was melted out, plenty of opportunities to start growing and begin the growing season for plants. And then the following year, when I kind of anticipated starting some field work in June, that field site did not melt out until about July 15th. And so these have really important implications to resource access for plants. So meltout timing affects how much water is available during the, especially in that early growing season, and also the onset of when the growing season begins. Another, though, precipitation driver occurring at the site is monsoon rains. So our field site is in Southwest Colorado, and every late summer, hopefully, we get a nice spurt of high intensity rainfall events linked to the monsoon. At some years, this has been really frequent and intense. And then I've had a couple years up there where I maybe got one shower and it was pretty weak. It's also been a challenging set of years for wildfires up there. I think right now as a field, we're just starting to understand how haze and reduced radiation from sun affects plants and their performance. But it's definitely a consideration in terms of when there's lower light intensity, these days did feel cooler than they might have otherwise. So that's just me being up at the field site and making observations over the last 6 years or so. But what does our kind of long-term climate data say? So there's a couple different ways to think about what these plants are experiencing. These are weather station data starting in about 1990s. 7 or 6, depending on the variable. And one way we can start to think about the conditions these plants are experiencing is how many days were really hot during the growing season. So that's days above 20 Celsius. If we just looked at my time up on Mount Baldy, we might think it's very hot relative to the recent past. But also, there was a lot of hot years in the early 2000s. Number of days between rainfall events has linearly increased over time. So in the early 2000s, a plant might wait maybe 2 to 5 days between rainfall events. And now it's much more common for 5 to 8 or even 12 days sometimes to pass between rainfall events in this community. But what does that mean in terms of total precipitation? Right now, it looks like there's not a lot of variation across years in how much participation is reaching these sites, even though a lot of these other variables like how much snow there is and how much monsoon rains there is. It seems like across the years, there's not a lot of across years there's variation, but the long-term trend is that kind of a comparable amount of rainfall and snowfall is falling at this site. We can look at that too through a PCA. So in the purple dots are all the years that we have demography data and have collected data at the Mount Baldy field site, and in the gray is weather station data where we don't have any intel about plant performance during that time. And my experience being up there, I would have thought, you know, we're seeing a lot of droughts, we're seeing a lot of years without snow, but really in the broader context of the last 20 plus years, we're seeing years that are similar to what occurred 20 years ago. It would be nice if we had data going about 50 years or even longer, but right now it just seems like this is a plant community that experiences a lot of environmental variation. Maybe some of those variables are increasing, such as the number of days between precipitation events, but overall, it seems like plants are accustomed to dealing with climatic variation. So that's my experience relative to weather station data. But what does that mean when you're on the ground? So when I visited these sites, and every year we take we take aerial photos that we orthorectify so that they're standardized across the different years. And these are all lined up so we can see how a single plot changes across time. And then the year is kind of picking up there in the top. And so there's a start. You can see some really lush years. You can see years where we pretty much had 0 plants in this plot, minus that really large guy over on the left. But depending on when you visit this site, and if you're making comparisons from one year to the other, your impressions about what this community looks like, the abundance and the overall performance of these plants across years can really vary unless you have several years of data. This holds for even looking at these broad demographic variables. So if you look at how many seedlings peak for that was 2015. We had the most seedlings out of in a year. When I came early on in 2018, I didn't know how to identify most of these plants because I didn't see any seedlings functionally until 2020. It was a real, it was a great year for me, honestly. But it just goes to show essentially, we haven't returned to total abundance that we had early on in this field project. But we are seeing some increases. It's hard to know if this is truly a community on the decline. It would have been interesting, for example, had we captured 2012, because that is an important drought year. And so only by extending this field project multiple years can we really start to understand how to link demography to the environment, and especially this environmental variation. So my kind of current framework and thinking as a graduate student has channeled more into this environmental variation direction, but also just how a plant experiences that. So one important variable for how environmental variation might be perceived is who are you hanging out with and who are your neighbors. So this is an aerial view of our Mount Baldy plot, and you can see it's pretty patchy. There's a lot of areas where there's no vegetation, and then there's it's hard to tell, but there's just clusters of vegetation. And then when you zoom in on that vegetation, you can see that they're pretty species rich, many individuals, and that's a pretty common looking cluster right there. So we do know from other systems what it means to have neighbors. So for example, in the Sonoran Desert, a classic interaction is between the saguaros and Palo Verdes. And the idea is that the Palo Verde can serve as a nurse plant for the saguaro by providing shade and through properties like hydraulic uplift. And also we know through Ray Calloway's work how the presence of neighbors affects whether facilitation or competition occur depending on elevation. So our field site's a high elevation site. And so if you looked at Ray Calloway's figure here, on the low elevation site, we can see there's not that much variation between control, meaning you have neighbors, versus neighbor removal, being alone. But there is a difference. It is a little bit better to have neighbors removed. But if you look at the high elevation site, your performance is much improved by having neighbors. And that's through these facilitative interactions between plants that occur at higher elevations. Interactions and having neighbors can also affect important community level characteristics, such as species richness. This is a study done in the cushion plants that occur in the southern Andes, and this showing that species richness within cushions is higher at the 700 meter elevation site and the 900 meter elevation site, but proportionately compared to outside cushions, the species richness is much higher at the higher elevation site. Okay, so going back to Colorado, what does it mean and how is your performance gonna depend on where you're growing? So if you're a seed and you germinate and start to grow, how is your experience gonna be different in an open landscape versus one of these hyper dense clusters with likely lots of individuals versus a slightly smaller cluster that maybe has fewer individuals to interact with? And these functionally are also questions about community assembly. What influences the species that we see in a community, and what are the mechanisms for species occurring in a community, and what distribution and what spatial arrangement? So looking at this figure from Hilaris Lamber's 2012, we have this idea of a regional species pool, all the different species that could be in a community, and then this concept of filters. So we have an environmental filter being that kind of first kind of hurdle a species would have to cross to be able to occur in a community, and then a biotic hurdle. So the idea of you're not being pushed out of a community, for example, through competitive interactions. And then what they're showing over here in these little pie charts is that interactions influence these environmental and biotic filters. What we don't know, what's typically kind of less considered in these community assembly mechanisms is dispersal. So that's that first actually hurdle in terms of a species occurring in a system. And one of the kind of concepts that I most want to explore in this time. So dispersal is that arrival and being in a community, and how does that interact with these other mechanisms of community assembly? So I organized my talk today in a series of insights, thinking about that title of how to succeed if you're a plant. And these are kind of this general, not ideas necessarily for success for an individual, but what we've learned over the last 5 years about what are the concepts that really affect plant performance in this system. All right, so the first insight, microenvironment modification and spatial overlap drive demography and species interactions in this Alpine community. My format for these insights is gonna feature the photos of co-authors below. So we have Rosalie Kappas and Oystein Opedal and then Benjamin Blonder. And so this work started a little before me in 2016 and utilizes data from this Mount Baldy field site, thinking about this factor of plants co-occurring, and then how does that affect their experience of the local climate around them. So our research questions, thinking about ways that plants can modify the microclimate. So we first asked, we know plants have this physical capacity to provide shade, and also through physiological properties can cause cooling and buffering of temperatures. So we first asked for question 1, how much are surface temperatures being modified? And then as part of that question, also how much is soil moisture being modified near plants versus away from plants? So soil moisture through mechanisms such as hydraulic lift, and also just in some cases higher humidity, the experience of water availability can be very different near versus far from plants. Then after addressing whether and the extent of whether microclimate modification occurs, and then the extent of that microclimate modification, we then asked how do vital rates respond to that modification, and whether factors about the plant influence that response. So aspects we're interested in is the size of the plants, the given year, and also neighbors and the density of those neighbors. I've talked about Mount Baldy quite a bit, but I wanted to give you its stats real quick. So it's in the Gunnison National Forest. It occurs in southwest Colorado, and it's located on a southeast facing ridgeline. The elevation is about 3,500 meters, and the site is set up as 50 2x2 meter plots. Every individual in this site is tagged, and what we do is an annual census during the growing season each year. For our demography data collection, since we've been working here for about 9 years now, we have an idea of how old many of these individuals are, or they're at least greater than 8 years old. We know the abundance of each species, and then we have vital rate data. So how well each species survives, grows, recruitment, and their fecundity. So for our microclimate data collection, just to assess if near plants versus away from plants, the microclimates differ, and thus if you had that neighbor, you might experience a different microclimate. We deployed lots of iButtons. So prior to my time in 2016, the crew deployed iButtons adjacent to 86 plants, and then also in 91 non-vegetated areas located at the corners of the plots. So these were deployed for 26 days. And then in 2018, I came along and replicated this study and deployed iButtons at 81 plants from 7 species. And I set this up as paired measurements. So just adjacent to a plant and then 10 meters or 10 centimeters away from the plant. You can see what that looks like over there. The iButtons were covered in parafilm and then wrapped in duct tape for waterproofing. And that also causes some reflectance to occur for the iButton. Soil volumetric water content was measured with a soil moisture probe. That was done in 2016. And so how that was done was there was a significant rainfall event on August 7th, 2016. And then so that reset the site. And then 2 days later, after some dry down and differential dry down patterns could occur, the site was measured. Soil moisture was measured at 3.8 centimeters depth. It was measured on 134 plants and 11 species. And these again were paired measurements adjacent to the plant and at least 10 centimeters away from the plant in a non-vegetated area. So I next will transition to the other variable I'm interested in for microclimate modification and what could influence vital rates, which is how many neighbors you have. So I at least suspected at this point that micromodification would occur, but I wanted a good way of estimating how much modification might be occurring. So one mechanism for doing that is just to look at how much overlap, physical overlap of individuals is occurring. So this is real data of polygons made for every plant in a particular plot. And from that data, we can see how much different polygons are overlapping and the identity of those polygons. And then I just made a little cheat sheet over on the right there to illustrate how this type of estimation is done. So each of the percent overlaps are relative to the individual. So if you look at individual A, only 25% of its area is being overlapped by another individual. And that individual that's overlapping with A is conspecific. And then in the case of B, for example, that has two heterospecific individuals overlapping with it, you take the sum of those two areas that is being overlapped with. Then finally, I also wanted to understand how overlap might differ above and below ground, since as a approach to understanding maybe how interactions also vary depending on the ecological realm. And so we had latitudinal root lengths that were collected. So the plants were dug up for about 5 individuals for many of these species, a few less for some others. And we could relate the size of the plant to how much its roots extended. And then I was able to make a linear function that would allow me to estimate size of plant versus its below ground extent. As you can see, for most of these plants, the bigger it is, the larger its diameter is below ground. Okay, and then finally, I just want to show you what all those variables look like. So for percent overlap, above versus below ground, you can see most individuals had relatively low below ground overlaps and above ground below overlaps. But there are some cases where you could have an individual overlapping with up to like 12 individuals completely. So some of these clusters are very dense, especially when you look, try to estimate how much species are overlapping below ground. Great. So for question 1, for most species, we observed buffered temperatures, so meaning higher minimums and lower maximums in plants compared to non-vegetated areas. So the red dotted line indicates the mean value for non-vegetated area. And you can see that for most species, we have cooler temps for soil maximum temperatures and higher minimums for soil minimum temperatures. We also, so we observed higher soil moisture in plants in non-vegetated compared to vegetated areas. And so for the figure on the right, we're looking for significant differences. Those are darker shaded boxes. And so for most, between species as well, we tended to see higher soil moisture in vegetation versus non-vegetated areas. Looking at those vital rates, I used a general linearized mixed model where I looked at growing season precipitation, the interaction between growing season precipitation, size, and interspecific overlap, and then growing season precipitation, size, and intraspecific overlap, the 3-way interactions between all of those terms. And then as random effects included the plot, so what of those 50 squares, and then the taxon. So for survival, we can see that in terms of some of the overlap parameters, on the left we have above ground, and then the right we have below ground models. We can see that survival depended on some of these overlap parameters as well as the interaction. But across vital rates, how much interspecific and intraspecific overlap affected vital rates really depended. So here's one where we see a strong effect of size and precipitation and indication of species interactions also affecting vital rates for survival. For growth, we don't see as strong of an effect of precipitation, but again, size was positively correlated with growth, and also some context dependency of when inter and intraspecific interactions are important for that particular vital rate. In this case, we saw that more for above ground estimates where that signal was lost for our below ground estimates. And then finally, for fecundity, I'm showing you probability of flowering, and then also how many flowers you have once you do, if you do flower. And again, that signal of this, the vital, that vital rates response is really context dependent, depending on the year, who your neighbors are, and your size. An unsurprising consistent trend of size being a positive predictor of this, and a lot of interesting, especially signals of interspecific overlap when competition has a negative effect on those vital rates. Great. I'm gonna clear this off for this talk, so I'm just gonna grab. Oh shoot, my phone is off. I'll turn it on. Great. So microclimate conclusions. Could I actually have a clock for someone, please? Yeah. It says I'm at like 60 minutes. Thank you. I don't want to keep you too long. Awesome. So the conclusions from that microclimate insight is that there's an important role of fine scale habitat modification in driving the spatial heterogeneity of vital rates. And this variation of how your vital rates are, whether or not you have neighbors, may affect these spatial clustering patterns that we see. Also, there's a lot of possibilities for how climate change may affect species' ability to modify microenvironments. We saw some years when, depending on precipitation, that had a large effect on how much soil moisture was increased in plants, and also how much temperature was modified across those 2 years. We also found effects of neighbors on battle rates dependent on the size of the individual and climate conditions. Great. So for insight 2, I then am asking, well, for insight 2 is that alpine plant species interact via wind-driven seed dispersal and seed retention. This project was done with a lot of different collaborators. In particular, I want to highlight the top center there, Emily C. The seed trapping work was part of her senior thesis. So working at the site, especially during the late fall, it's really easy to see that there's lots of seeds collected around these plants. So we have this idea from our first insight that microclimate modification is affecting vital rates and promoting clustering, but it also really seemed like there could be a physical element also promoting this co-occurrence, and this through physical trapping and retention. Looking at earlier research, there have been previous studies exploring this idea of seed trapping. This is a 1997 paper by Aguiar and Sella. And so they looked at how many seeds were available through seed pores in grass, shrub, and then comparing bare soil close and far. And they found that depending on the microsite, the number of seeds varied, but that in bare soil far away from vegetation, they didn't find any seeds. And so that opens this idea of seed dispersal as a mechanism of species interaction. So in this kind of conceptual diagram, we have the idea of a parent plant dispersing, and it could encounter a plant and not be trapped or dispersed or retained and go along its way. Also, potential option is that the seed could encounter a plant, be trapped, but ultimately not be retained. It could blow off or maybe pushed by rain. And then a third option is that a seed could be trapped and retained. And the probability of these different interactions could depend on the plant traits and the seed traits. So what I set up over here on the right is just a schematic of how that could look. So trapping is in red and retention is in blue, and darker colors indicate that interaction being more likely. So one possibility for the yellow plant is that it's really, regardless of the seed, it traps everything, but has low retention equally well. A plant could be unlikely to trap something, but have good retention. And also the interactions could vary depending on who the seed and the plant is. So we asked in our site whether, to start off, do seeds really vary in their seed functional traits? Is there high functional diversity occurring at the Mount Baldy field site? And then do we see different probabilities of seed trapping and retention depending on who the plant is, their identity, and who the seed interactor is? And so we set up 2 different experiments. So this is, you can see with the orange dots that those indicate the 4 outer corners of our census plot. And then we did an experiment where we tested seed trapping using a portable wind tunnel that we built and blew seeds into the path of the plants. So that's those locations where we did that work is represented by the little teal crosses. And then we did a second experiment of seed retention where we inserted seeds into plants and monitored how long they were retained. So that's the blue X's. So we had, for these projects, we used 6 plant species and 6 seed species. The plants were selected because they were the most abundant at the field site, but they also capture a lot of the functional diversity at the site. And then the seeds, although they don't correspond to the most abundant plants, they were the most abundant seeds available during the year of collection. And similarly were selected to capture a lot of functional diversity. So to start off in addressing question 1, we had to measure seed functional traits. So for all of those species, we measured at least 100 seeds from 10 seeds from 10 individuals. And then we did a principal component analysis to look at that functional overlap of the seeds. What we found is that for many of the seeds, there wasn't a lot of functional overlap. But for seeds with the pappus, which is the pink, teal, yellow taxa, those taxa are similar along PC1, but diverge along PC2. I wanted to really highlight those 3. They're going to appear in a lot of the other figures. And so how this slide is arranged as a little how-to guide for the next couple slides, all the plant species are color-coded. You don't have to remember the colors, but it might be a helpful mnemonic. And to think about these different seed taxa, I think it's useful to think of the first 3 as differing in size from big to smaller along PC1, and then those bottom 3 representing the pappus trio. Okay. We'll return to them, but I just wanted to highlight that. All right, so we built lots of wind tunnels. Our vision of what makes an effective wind tunnel is one where there's laminar flow that was consistent throughout the tunnel. We gauged that using a hot wire anemometer, mapped the whole area of the tunnel, and then also for whether the flow was laminar, just held a piece of yarn in the tunnel to make sure it wasn't swirling, but just flew straight back. So this is my favorite. I call it the staghorn. And ultimately, we decided on this particular wind tunnel that it fulfilled all of our qualifications. So we fluorescently dyed seeds of those 6 taxa, and we set up an experiment where for each species, we blew 5 individual seeds into the plant and looked at the proportion that were trapped. We had 10 focal plants, 5 from Heterotheca villosa, and then 5 from the Lupinus auranteus. We chose these 2 taxa because the lupin is a lot less dense at its base, even though it does have a lot of vegetative structure up above. Is lower ground density of vegetation. These taxa are also both really abundant at the field site. So essentially, it's kind of like a walking off the plank, essentially, for these little seeds. They went on a little launch pad platform. We had the wind speed about 5 meters per second, and monitored the proportion that made it to the back of the tunnel and didn't get stuck in the plant. So seeds that were considered trapped are in the top left. You can see the neon dye on it. So any seed that was underneath the plant was considered trapped or physically touching, and then if it made it into the net in the back, not trapped. I then analyzed the proportion trapped relative to the plant that is being blown into, the identity of the seed, and then also how big the plant was. And I found that larger plants are better at trapping seeds. For comparing the two plants, Heterotheca villosa and Lupinus arataeus, we do see some differences, especially at larger sizes. Those did not end up being significant. There's potential with more replication. We could, and also a larger range of plant sizes, we might find some patterns. But what's really interesting is that the trapping did not vary among the seed taxa. Similar trapping across all of them. The next part of our understanding dispersal as a species interaction is to look at seed retention. So that's that physical insertion of a seed and tracking how long it stayed. So for that, we deployed nearly 3,000 seeds into 83 plants, 6 seed species, and then checked how many of them remained after 2 days, 4 days, and 11 days. So that process also involves a lot of fluorescent dye. This is just the powder you use at like a holly celebration. It gets dusted on, and then we can individually track different seeds in different groups. Those got inserted directly into plants, or for the paired non-vegetated area, 30 centimeters away, and then we track them over time. Luckily, with that fluorescent powder, we were able to see them. Many seeds lodge themselves in between the scree, and sometimes we had to use black light. But what we found in terms of whether a seed was dispersed, that really depended. So we looked at whether a seed dispersed based on the microsite. So that's whether it's a non-vegetated or the identity of the plant. And the interaction with that with seed taxon, the interval duration, how much time between the check, and then as random effects, the check. So whether it's the first time we looked or the second. Plot type, this indicates vegetated versus non-vegetated. And so these plots, once in their full complexity, feature a lot of colors. So the intro to them is just to compare Eriogonum undulatum or buckwheat with Senecio crassulus, a little aster. So here's again where that color coordination, same, the orange is always Eriogonum, whether it's a seed or a plant. So Eriogonum is a very dense plant, and what we saw is that its retention, the probability of seeds dispersing after 8 days was a lot lower than the probability of seeds dispersing in the less dense plant, the Senecio. Dispersal likelihoods were similar between Senecio and the non-vegetated areas, and the seeds with the pappus, which is a Senecio, also were more likely to disperse by the end of the experiment. Great. Looking at our focal taxa again, so these are the 6 species that we studied, ranging from grasses, the asters, and then these are seeds also in order of PC1s. So the seeds without a pappus essentially, and then seeds with the pappus. So what we see is that seed retention varies depending on the host plant, the seed taxon, and then but there's a lot of interactions between what seeds are retained and what depending on what plant they are in. So Heterotheca villosa, for example, retains seeds similarly well, regardless of what seed type is in it. And then other plants like Senecio crassulus, which has pretty high dispersal. It really depends what seed is in it and how well they are retained. So again, that teal, yellow, and pink are those seeds with the pappus, and those are really most likely to be dispersed. How that looks depending on the principal components, we see that what principal components, how they influence dispersal depends also on the microsite. So we can see for open areas like non-vegetated areas and plants that are less dense, such as the Senecio crassulus again, we see an influence of these seed traits on retention. But for plants that are more dense, like that yellow Heterotheca villosa and Ipsia gordonii in the blue, seed traits have less influence on retention. So conclusions, seed trapping and retention are a form of species interaction, and they depend on the physical attributes of the plants and the seeds. In kind of considerations for why this matters for community assembly, a lot of these systems where we see abiotic dispersal, such as grasslands and alpine, are also areas of a lot of community shift and change. Things like presence of cattle, increased grasses or shrubbification can affect these dispersal processes and the likelihood of seeds ending up in certain microsites. And incorporating seed trapping and retention mechanisms is important for understanding community assembly. So this is, we're nearly at the gate. This is just a quick little teaser. I wanted to just introduce my third insight, and one that I more invite you to ask me about after the talk, in case you have any questions, as the work is preliminary. But so this is work that I'm doing in collaboration with Rob Sverro Gomez, and we're looking at what other mechanisms can plants use to respond to environmental variation. So one way that plants can do that is by allowing their vital rates to either track environmental variation or allowing their vital rates to be stable in response to environmental variation. And critically, it's what the vital rate we're considering in those comparisons is the one that most contributes to population growth rates. So, for example, we often compare plants in terms of whether they have a fast or slow life history, whether they reproduce many times or a few times. And another point on that axis is how their vital rates respond to environmental variation. Questions I'm interested in addressing is whether plants that are reproductive, so in a more adult stage of life, whether they are more likely to buffer, meaning their vital rates don't vary, and also long-lived plants, whether they are also more likely to buffer and not allow their vital rates to vary in response to variation in the environment. Yeah, please talk after if you're interested. And then I just want to circle back to my title. So how to thrive in a community, a plant's practical guide to dispersal, and then revisit this idea of the filters of community assembly. What my work shows is that it's really hard to put these filters down sequentially. We have, from our insight 2, we see that dispersal can be a mechanism of species interaction. From the first project where we looked at microclimate modification and neighbors, we found that those interactions really also depended on the climate. And so that biotic interactions and environmental filters can also interact and occur simultaneously. My advice for a plant is to just do your best. So I wanted to thank everyone in ESPM, especially for having me as your distinguished speaker. I moved here halfway or so through my PhD and found a lot of community, especially through recreational sports. We don't have the 2020 softball league photo because that season ended early, but the Tree Huckers Frisbee team is the best, and we're also made it to the playoffs every time. I had a really wonderful qualifying exam community, and what I like about the community structure in ESPM is just the opportunity to interact with faculty with very different outside research. So Albert, for example, and I spent a lot of time discussing dispersal and aquatic systems, and really helped me shape how I think about meta communities. And then I've gone to Laval's about 80 times as a grad student, and every time it's great. Yeah, I also want to highlight the Ackerley lab. They've been my second lab while here. Again, just running beta breakers. It's great that like ESPM can come together and form this like a non-official team for legal reasons. And then also all the grad support staff have gotten me out of a lot of pinches. And just my grad student support network. Kendall, Lauren, and Jesse have been there for helping me write a lot of proposals and thinking through life decisions and research decisions, and both have been really valuable. I wanted to just point out that many of these really nice remote sensing images were made by Ian Breckheimer. He's our Rumble collaborator. And I also wanted to thank Ben, who I think has told me to be safe also 80 times.
Go.
Yeah. He's really good at hopscotch. And he is a bulldog, if you insult Aspen. So it's important to keep that in mind with your interactions with him. Yeah. And then thank you, everyone.
Quick time for questions for Courtenay or anything else you might like to share with her.
I was wondering if you noticed any like morphological features of either the seeds or the plants that impacted retention or dispersal.
Yeah, so the retention was higher in plants that had higher vegetative density. And then also it seemed important like where vertically that vegetation density was. The plants that had more density towards the bottom versus up top retained more seeds. One of our future directions is to try to like quantify that. It could just be through some sort of estimate, basal versus top, but I think there's also potential for like doing remote sensing to try to understand these 3D shapes of plants. Oh yeah, just
I noticed in your pictures of your field site, there seem to be like very strong edges in habitat. Like there's like lush, like fully vegetated meadows. And then all of a sudden it becomes this kind of rocky, like there's this little patchy vegetation. And I was wondering if why does that happen?
yeah. So our field site is on very poor soil. It's essentially about 10 centimeters of scree on top of bedrock. And then the surrounding areas have much more developed soil.
With the talk, I was thinking a lot about how, as these species are moving altitudinally with changing climate, how the neighbors were there when they first dispersed to a new patch have a really big impact on weather rates. So having a sense of this and all this time, do you identify species or species characteristics that you think are more vulnerable to sort of being beat to the next spot up the mountain? Or is that maybe a really simplistic worry about what species might be vulnerable?
Yeah, it's a great question. So I think probably vulnerability could be indicated by not very trappy and not very retainable. And so there are seeds that are, but because all the seeds can be caught in some plants, I think what would end up happening is the microclimates experienced by the seeds that only can be caught in some plants might vary. I really have a lot of curiosity about fireweed that seems to be encroaching in the area. And it's a papist seed that's very cottonly. And so currently abundance of fireweed is 0, and then we'll talk in 2035. And then also where it ends up is going to be an important question.
Thanks.
Okay.
Yeah, make it more really awesome to see the results of these chapters. And thinking about chapter 1 and 2, how variable the vital rates were, and the variability seems to be given by local scale factors. I was wondering, is that good news in the context of climate change, meaning that, microhabitats and microclimate needs to be matter, matter more than, like larger scale, like increases in drought or something like that, in the landscape at a larger scale.
Yeah. So it's a great question. I think we're still seeing large scale declines in this community, regardless of having neighbors or not having neighbors. But plants that do have neighbors and do occur in these clusters seem to be holding on longer, is kind of my current perspective on it. And it makes me wonder too, if like those plants that occur in clusters might be better set up for responding to environmental variation. So perhaps, well, this year might be very good due to our snowfall. And so it might be that we could see good response in the next couple of years that could help stabilize this community. And the plants in clusters who survived through some drought years might be best poised to do that. I think many more years are needed.
So very cool. Not being a climatologist, how do you say, I'm thinking about the slide you showed about the relative importance of filtering and other competition. How would you say your system is somewhere different from, say, what happens with those processes in other California ecosystems like Denver's or Chaparral?
Yeah. So that's a direction I'm interested in exploring for my postdoc. So the that climate figure where I show the number of days between precipitation events, that it's increased over the last 20 years. That's a phenomenon that's occurring across the West. That precipitation is rare, but higher intensity than it used to be. And so that I think is really applicable for thinking about California annuals that occur in the desert or in our grasslands here, where the timing of that precipitation affects germination, but how much rain they get in the intervening time really affects how long those species can persist. I've talked with some folks who work with annual species with greenhouse experiments. And it was we were trying to discuss how long a project should last. And they were saying just like, if you keep on watering them, they're not gonna die. I think a lot of what makes them annual is that cessation of rain. And so I think thinking about that kind of climatic variation, I think there's a lot of applicability to California systems.
I kind of want to ask you about like an overall ecosystem functioning, but it seems that your conclusion is that the life of these plants is very complex but looking at your like second piece of data where the retention of the seed, it seems that the two species that were retained in where there was no vegetation were very low in kind of landing under a plant. So have you looked at these responses by grouping plants, for example, like instead of using individual species that make things very complex,
Yeah.
grouping plants that are the ones that land in no vegetation areas compared to the ones that are adapted to use like a kind of a nursery plant?
That's would be a really good way of looking at it. I think you could do that by comparing pappus to non-pappus seeds really easily. And then asking, one, if you just group that, do some of the pattern, are there different patterns than we saw at the species level? You mentioned ecosystem, and this is still a community scale, but one thing I do want to do for a future analysis is try to link these retention and trapping probabilities to the extent distributions of like the living plants, to see whether plants that have mutual high trapping and retention with their seed buddies, whether they co-occur in the landscape.